I guess one common misconception of the microbiome is that all bacteria are the same and they all work the same way. At a fundamental level, that is true, but realistically, they're all specialists. So, one way to really move forward in this field, and it's going to take a lot of groundwork, is to figure out what each of these specialists can do.
Hello and welcome to Swine Podcast. I'm Dr. Tara Donovan and my guest today is Dr. Benoit St-Pierre. Dr. St-Pierre is an associate professor in the Department of Animal Science at South Dakota State University. Welcome to our show, Dr. St-Pierre. How are you?
I'm doing well, thank you, and thank you for the opportunity to be on your podcast. It's my first time on a podcast. Maybe to start with a little bit about myself. I am French Canadian, so English is my second language. When I get a little nervous, sometimes my accent will come out, but I try to keep it under control as much as I can. I did my bachelor's and master's in Quebec City at Laval University. And then I did a PhD in cancer research at the University of Toronto. And I decided to take a turn towards working in sustainability. I figured that there were more people dying of hunger than people dying of cancer. So that was kind of my, I guess, a self-reflection on where to pursue my career. So then after cancer research, I decided to go into plant molecular genetics. And then while I was at the University of Vermont, I met Andre Denis Wright, who is one of the leaders in methanogen research. And he had just moved to Burlington. He was looking for a postdoc, and that's how I got into the microbiome business. And the reason why I was able to switch from cancer research and plants and microbiome is fundamentally my expertise is in molecular biology. So that's how proteins are expressed from genes and how that expression is regulated. So since all organisms have DNA translated or transcribed into RNA and then translated into proteins, I was able to make that switch. But I'm very happy in the microbiome world. When I started in this field, it was just at the right moment when it was taking off, taking advantage of new technology that was available. That was back in 2011.
Explore MetaFarms for all your livestock production data needs. Experience our integrated software platform offering complete operational visibility from breed to market. Utilize customizable dashboards for data analysis and benchmarking. MetaFarms mobile apps enable offline access, maximizing efficiency and enhancing profitability. Visit us today at MetaFarms.
Perfect. Thank you for that introduction. That's great. And definitely an area that's important in livestock production. So we're going to talk a little bit about how kind of our lives overlap here in just a bit. So tell us about, let's just talk about the microbiome. Okay. And, so get us started a little bit. How does it work?
Well, it consists of, depending on the gut environment. So if you take fish or shrimp, for example, they might have a few dozen types of different bacteria in their gut. If you go into swine and humans, estimates will vary, somewhere between in the hundreds to the thousands. And then if you look at ruminants or herbivores in general, you get into the thousands for sure, and possibly into the tens of thousands. So one of the reasons why there's so much diversity depends really on the diet of the host mostly. Bacteria are typically specialists, and they're not able to do everything on their own. So they depend on other members of their community to help them out and give them substrates that they can't metabolize, and themselves in terms of substrates that they can use is also very limited. So for example, if you have plant fiber, it will be cellulose, hemicellulose, pectins, and possibly starch also as well. So there are very few organisms that are able to digest all of these or utilize all these different types of substrates. So the diversity in the gut comes primarily from all the different chemicals or different substrates that are part of the host's diet. And fundamentally, and it's really hard to define what is a healthy microbiome. We know what a diseased microbiome is because it's invaded by pathogens. But in terms of a healthy microbiome, my definition would be it's essentially communities of bacteria, so specialists that can eat things that the host is unable to digest. So those would include cellulose and hemicellulose and pectin, for example. So that's the basis of the microbiome, where you have a wide array of bacteria with different metabolic capabilities. And it's not that they hold hands and sing songs together, but they all work well in terms of complementing what they're unable to do themselves.
That's a really good description. And so let's talk a little bit about. So you mentioned that ruminants have more species or varieties than pigs. And so since this is the Swine It podcast, let's dig a little bit into pigs and talk about what's special about them and what do you work with in the pig world.
Well, I do this kind of work in collaboration with nutritionists, with swine nutritionists at SDSU. The advantage I have is there are many of them, and they work on many different aspects. So it goes from sows and producing piglets to nursery pig diets, and then later on to grow finishing phase. So I've had collaborative projects on these different stages. And what I find in general is if I think something is not going to be that interesting, it turns out to be way cooler than what I expected. So I'll give you an example. So for me, nursery pigs are the most fun because the transitions that happen cause major changes in the microbiome composition. And when you're a basic researcher at heart, like I am, seeing big changes like that, you can relate to function, and that's really cool. When I participated in it was a study comparing heavy market pigs with pigs that were much lower in weight. And I thought, well, it's probably not going to be that much difference. But it turned out that they were different. And not only that, but the bacteria that were prominent in the hindgut of these animals were completely unknown. So one of the basic assumptions is that probably everyone's heard of lactobacillus, and lactobacillus have magical powers that keep us healthy. And you would have assumed, based on what we know, that lactobacillus would have been the more prominent one. But it turned out that these unknown bacteria were the most dominant. So to me, what gets me excited about research is the opportunity to discover new things, basically, and that's why it's a never-ending playground for me. And I'll probably can work until I'm 150 if I can handle it.
So, what I understand about the microbiome, and you mentioned we're talking about pigs, and a young pig goes through some stressors, right? The weaning event is a big stressor. So, do you work in the area of how stress or some of those chemicals affect the microbiome?
Not yet. Well, one of the very tricky things with working with animals is there are a lot of factors that are involved. There are a lot of events that happen. Most of them we don't control. Um, the One of the challenges with stress is it can be a response with very low hormonal input, and depending on the type of species, they may not be responsive to them. So it's really hard to control that type of environment. It's really hard to control the effect that stress has um, on this particular aspect. I've focused mostly on um, big or conditions that are very different. So, for example, the change in the different phases of a nursery diet. So each phase will cause big changes in microbiome composition. When you start looking at very presumably subtle effects, like, for example, a pig being stressed for maybe 4 to 5 days, that can be really hard to tweak out in an experimental setting.
Oh, that makes sense. Yeah, that makes sense. So, what are some applications that you're working on, or some of those collaborations that you do? So, it sounds like in the nutrition side, looking at the different microbiome response to different feedstuffs and diets. So, what kind of applications out in the pig world are you working in?
Well, I haven't worked directly in applications. I've been involved in projects that were testing commercial products to see if they had an effect on the microbiome. And those were also opportunities to, if you look at the control group, people usually overlook the control group, but when you're looking at um, the microbiome, which is really an unknown world, it can really reveal a lot of basic information about what's going on. Um, in terms of progress, we're currently trying to build genomes for these bacteria. And through genome building, we can figure out what they like to eat. And by knowing what they like to eat or what they need or what they don't like, then we can move on to try to modulate their activity. So, one example that people are probably familiar with are prebiotics. So, prebiotics, the way they work is you essentially feed um, good bacteria things that the host is unable to utilize. So, if we can figure out which bacteria are good for the pig, and then we can provide a prebiotic in the diet, then that will promote the growth of these good and healthy bacteria. So if I can go back for a second to the what I mentioned before about the heavy market pig study, where we identified these really prominent bacteria that were unknown, we compare them to their status in nursery pigs, and they're present but at very, very, very low abundance. So presumably, if we can figure out what these unknown bacteria like to eat, and if we can put that in the diet of nursery pigs, maybe we can get them to a more mature status faster. So that would be one application, but it's not something that's going to happen in the next few months. It's going to take a little while before we get there, but that's kind of the concepts that I like to follow.
Okay, that makes sense. So, kind of, I would manipulating the intestinal microbiome to adapt in either growing faster or what we would see as a different phase of life for them. So, really interesting.
Yeah, and then also, I'm not sure about growing faster in terms of nutrition, but most certainly be able to increase disease resistance. So presumably, an adult pig would be better equipped to resist disease as opposed to a nursery pig. So if we can get them faster to a more mature state, then hopefully that can help them, let's say, focus on the growth side and not so much on resisting pathogens.
Yeah, really interesting. So, what about you talked about sustainability kind of at the very beginning of our conversation? How does the microbiome fit into the overall sustainability in livestock production?
Well, there are different aspects. One of them could be nutritionally. Um, Pigs will typically have a soybean meal and corn based diet. Um, in the future, it might be better to feed them biomass or things that humans are not able to digest. So we could potentially manipulate the hindgut so that pigs can be better at utilizing biomass or more fiber-rich um, uh, diet ingredients. Another aspect that's often overlooked is manure management. Um, and bacteria can be utilized to control odor. We can also increase. I know that methane is really supposed to be really bad, but if you actually harvest it from manure pits, it can be used as a source of energy, and there are some applications of that already. So that's one part of the microbiome that can also be utilized to maximize, in this case, would be methane production.
Yeah, absolutely. Definitely an area that, the odor and the effects on the environment are kind of hot topic, right?
Yep.
So what are some future projects that you're working on and your vision as you see in the space that you work in, application for us out there in the field?
Well, as I mentioned before, there are a lot of unknown. I guess one common misconception of the microbiome is that all bacteria are the same and they all work the same way. At a fundamental level, that is true, but realistically, they're all specialists. So one way to really move forward in this field, and it's going to take a lot of groundwork, is to figure out what each of these specialists can do. And that's why I'm really focused on genome building of known and unknown species. And then based on these metabolic activities, then we can say, well, if if this particular species like to use oligosaccharides, or maybe it can digest mannose, then we can feed it yeast wall or yeast cell wall or some of these types of products. So that's where I really see future applications, is figure out which are the ones that are the good ones, and then how to keep them happy. And keeping them happy basically means to give them what they like to eat. And prebiotics would, in my opinion, be the best way to go about this. Maybe one more note on, we hear probably more about probiotics than we hear about prebiotics. The challenge with probiotics is most of the species that are available are not native to um, either the pig gut or even the human gut. And trying to develop species from the gut into probiotics it takes a really long time, just in terms of not just developing the product, but getting approval from the government to be able to utilize them. So that's why prebiotics, again, in my opinion, would be the better way to go, because you don't have to go through all these hoops, and it's most likely a more straightforward way of getting to the same result.
Okay. So let's expand upon that just a little bit. So, basics. What's the difference? Is the difference in the application or the type, or the difference between a prebiotic and a probiotic?
Well, the concept the concept of a probiotic is you're adding a living organism into, in this case, the gut, and then that organism, by its function, by proliferating, is going to be beneficial, and then that benefit could be from eating something that the host is not able to eat, or produce volatile fatty acids or short chain fatty acids. The downside with that, and I think that most probiotics are just food for other bacteria in the gut, because they're unable to survive. The flip side, which is the prebiotic, is to take advantage of bacteria that are already present in the gut. They're already well established. They have function that fits in, and it's just a matter of making sure that they stay happy and that there are no fluctuations. Um, one of the currently one of the concepts in terms of developing diseases is dysbiosis. And then dysbiosis is an imbalance in community. So, the good bacteria are not happy. They go down in abundance. And then the bad players go up in abundance. And then there's a transition phase where it could either go back to being good, or it could further devolve into becoming a disease state. So the addition of prebiotics would be with the goal of making sure that the good bacteria that you want to keep happy remain happy.
I like it. That makes total sense. And so you really are working in very, very I would say fine detail in terms of the microbiome, trying to understand each of those players and what, like you said, makes them happy. So,
Yep.
Really, really interesting. So we're going to kind of complete our time together, our discussion. So what are some final thoughts or take-homes from today that you have for our audience?
Not all bacteria are created equal. I think that's one common misconception about the microbiome. So just not any bacteria can be an effective probiotic, for example. The other take-home message is not all bacteria are bad. We tend to think of bacteria as germy and icky and causing diseases. In most cases, the vast majority of them are either or just have no effect on us whatsoever. And even though there's similarities with animals, they're not mini animals. They are, I would compare them to robots. They are designed to do one thing, and that is to divide, make copies of themselves. And if you want to keep the ones that are basically the ones you want to keep, then you just have to make sure that you know what they like to eat and give it to them.
Very good. I like those, and you're right. I do really often think about bacteria as being bad. I'm a veterinarian, and so automatically I go to let's let's kill them, right? And so
Right.
it's a different concept of utilizing the good bacteria to make the pig healthy.
Well, my sister-in-law invited me to her class through Zoom a few years back, and the reason she invited me is the students did an experiment where they took swabs of different areas of the school, and then they streaked those on plates, and then she was panicked and disgusted by what was growing on the plate, and I just told her that they're probably not harmful at all for humans. Um, they just grow where they find their food, basically. So that again is one common misconception about bacteria.
Yeah, that's a. I remember doing something similar to that in microbiology class. So no, really, really good. Well, I appreciate learning about what you do and the connectedness that researchers have to veterinarians, and so it's been really great to understand your area of expertise. So, but before we completely end our time, I want to ask you the three questions that we ask all of our guests, just to get to know you a little bit better and to stimulate some thoughts on our end. So,
it's time for our famous three. Why risk it? Your herd may have antigenic gaps that are not being covered by the products currently available. Custom-made vaccines provide a unique advantage in that they can be formulated to a herd's specific bacterial and viral needs. Choose the exact isolates, adjuvant, packaging, and size you need with Vaccinova for a vaccine formulated to your needs. Find out more by visiting us at vaccinova.us.com. That's VAXXINOVA.us.com.
what's your favorite book or resource that you use in your area of expertise?
I am not a fan of books because they're just not up to date for for what I do. The internet is an amazing resource of information for peer-reviewed literature, and that's really what I use mostly. It's very seldom that I would use textbooks, even for designing my courses. I just go straight to peer-reviewed literature. So I
Yeah.
guess I didn't answer your question properly.
Oh no, you did. You did. It can be a resource. Exactly. I said book or
Yeah.
resource. So
Okay.
absolutely. And so how about something that you would recommend we look into a book or resource outside of your area of study?
Well, I would say that probably the most influential book that I read as a biologist was about the Gaia hypothesis from James Lovelock. So I don't know if anyone is familiar with that book, but the concept is really intriguing in thinking of Earth as a living organism and with regulatory systems. And it's a very elegant model of how our planet works. And the other thing that I thought was cool about this is Dr. Lovelock basically came to this hypothesis by working, I think it must have been with NASA, but they were trying to figure out ways to detect life on other planets. So that led to, you know, what makes a living organism basically, and then how do you recognize them? So Gaia hypothesis I would recommend.
Okay, really intriguing. And I have not heard of it. So thank you for that. And so the last one is share with us an attribute or characteristic that you feel sets successful professionals apart from others.
I think admitting to yourself that you are wrong. And at least as a researcher, that's really important. Um, I would say that there are always two types of people. So let's say there are two types of researchers. So they're the researchers that only believe in the theory that they come up with. And then experiments are essentially, they're successful only if they validate whatever model or theory that they followed. And then on the other hand are the one like me that say, well, okay, well, I was wrong again. It's just another day at the office and we'll just follow the evidence. And I think that probably applies to a lot of other professional fields where no one likes to make mistakes and no one likes to admit to making mistakes, but I think it can lead to a lot more challenges if you're not honest with yourself and say, okay, things didn't work out, I was wrong, or I made a mistake, and then move on from there.
Yeah, very good advice. Very good. Well, thank you so much. It's been wonderful having you on the show. And until next time, have a great day.
Perfect. Thank you very much for the invitation, and it was a pleasure on my end as well.